Hello, my name is Hannah Phillips, and my project looked at the comparison of different ionic and molecular chemicals as a means of controlling harmful algal blooms. Cyanobacterial harmful algal blooms occur in freshwater environments when blue-green algae grows in eutrophic conditions and begins to produce toxins. This can cause extensive economic and ecological damage in the environments they occur in. It can be extremely expensive to try and remove these blooms, and it also wreaks havoc upon the ecological systems surrounding them. Currently, there are very few methods to control or prevent the growth of cyanobacterial harmful algal blooms, and there are currently no methods available that address toxin production by cyanobacteria. The organism that I've observed in my experiment is called Microcystis aeruginosa. It is one of the major contributors to cyanobacterial harmful algal blooms, and it is specifically concerning because it produces a toxin known as microcystin. The goal of my study is to examine the delivery of different chemicals to try and make it stop producing the toxin. We use calcium chloride to make the cells permeable so that it can uptake a different chemical called an ASO treatment, or an antisense oligonucleotide. And the way this ASO treatment works is to disrupt the process of the central dogma. In the central dogma, DNA is transcribed into RNA, and then the RNA is translated into proteins. And the way my ASO treatment works is it is designed to be complementary to the RNA strand that produces microcystin. So when it is delivered into the cells effectively, it will bind to the RNA that produces microcystin and prevent it from producing the toxin before it actually happens. So in my experiment, we observed 4 different treatment groups. The first was the control group with just water. The second had just the ASO treatment. The third had the ASO treatment plus calcium chloride, and the final group had just calcium chloride. The treatments were observed over the course of 72 hours in order to relate cell counts to the amount of microcystin present in each sample. All of my cultures were grown in VG11 growth media over the course of 7 days. Every day, samples were taken in order to directly count the cells in each sample through direct enumeration as well as through absorbance values. And they were also saved for the final day's test, which was the ELISA plate. And the way the ELISA plate works is in each well present on the plate, there exists a predetermined amount of microcystin analog with an antibody conjugated to it. And it is a directly competitive test with inversely proportional results, meaning that the more color that is present in the plate once it's done, the less toxin that is present in each sample. So after we got the results from the ELISA plate, we were able to manipulate the data to generate a standard curve in which we were able to determine the total concentration of microcystin present in each sample from each culture. Afterwards, we related the amount of concentration to the absorbance values. And that paints a picture that says that the microcystin concentrations were steadily decreasing over time. That is what we hoped for. But when we related our results to the direct cell counts, it tells a completely different story. It shows that the toxin production began to increase in the last 72 hours for all of the samples. But what is interesting is that the treatment with the antisense oligonucleotide and calcium chloride, it does appear to have the lowest reported toxin in just 24 hours after treatment. But what is concerning is that it ultimately begins to spike again in the remaining 48 hours. And what is especially concerning is that the treatment group with just the calcium chloride appears to spike more than all of the others, indicating that calcium chloride is increasing the production of microcystin. This is absolutely counterintuitive to what we were hoping for. So in conclusion, when calcium chloride is administered on its own, it is not a suitable agent for controlling the production of microcystin in each cell. But it does appear that the combination of calcium chloride with the ASO treatment will result in lower microcystin production than some of the other treatments. But we do need to look at other methods of delivering the antisense treatment to try and discover a way that is more effective and does not increase the production of microcystin. We also discovered that the absorbance values are just not specific enough to the cells in order to relate an accurate count. They just are not specific enough to Microcystis aeruginosa. Further studies should be conducted to observe the timeline of microcystin production in order to observe the time frame in which microcystin begins being produced in the extracellular media instead of just within the cell. Further acknowledgements, special thank you to Dr. Lahiru Jayakody for letting us use his lab's plate reader, as well as Dr. Derek Fisher for letting me use his multi-channel pipette. Thanks to the SI Bridges program for allowing me this opportunity, and to Dr. Scott Hamilton-Brehm for assisting me throughout this entire experiment. Thank you.